We are Only Oncologists in the building. I'm Dr. Shruti Patel. I'm a medical oncologist out in the Bay Area. And today I'm going to be talking about mRNA vaccines and kind of how they're relevant in cancer care.
Dr. Emil Lou from the University of Minnesota. I'm a medical oncologist, and today I'll be talking about gene testing for safety in chemotherapy.
And I'm Dr. Shaalan Beg. I'm a medical oncologist from Dallas, Texas, and I'll be talking about whether AI is about to replace your physicians or not.
Looking forward to this discussion, guys. So, Shaalan, tell us a little bit about what you want to talk about today.
Yeah, I've been following the impact of artificial intelligence on clinical care. There's a lot of discussion that's happened in terms of how AI-based tools and informatics-based tools are going to impact. Are doctors going to be out of their jobs? Is clinical care going to become better? Are we going to save money as these tools start becoming implemented in the clinic? There was a very high-profile paper that was published by the team from Microsoft. It's a preprint published by the team in Microsoft, but it got a lot of traction in the mainstream media and on social media, which showed that an artificial intelligence agent powered by Microsoft, compared to physicians, had 4 times the amount of diagnostic accuracy than physicians at 20% lower cost. So that sounds great, right? So now we have AI tools which may be able to do a better job than physicians. So I wanted to take a deeper dive into that study.
I mean, that sounds crazy. Obviously, every headline we got to know the deets of the study. So maybe tell us more. What kind of? I guess I'm kind of intrigued about what kind of diagnostics they are. Who were the people that they asked? Who were the physicians? Yeah, what were the cases?
Yeah, so what they did in this study is that they took 300 cases from the New England Journal of Medicine's CPC series. And as a lot of our listeners are going to know, the CPC series are the cases that are diagnostic challenges where at Mass General they have seen an interesting case, a complicated case. They put that together in a way to challenge the readers to think about sometimes obscure diagnoses, but challenging diagnoses, and then it's an opportunity to educate people on the specific topic. So for this study, they took 300 CPC cases that have been published in the New England Journal of Medicine, and they compared the performance of a couple of different artificial intelligence tools with the performance of the physicians. So the AI is the MAI Diagnostic Orchestrator, and this is a virtual physician, and they trained 5 different agents. So you have the doctor hypothesis agent, the agent that is going to ask questions and come up with a list of potential diagnoses. They trained a different agent that challenges those hypotheses so that only the ones that survive will continue to bubble up. A stewardship agent whose goal was to come up with cost effectiveness and to put in a layer of cost effectiveness on the decision making. Doctor checklist, they call it the doctor checklist just to make sure all the major elements of coming up with a diagnosis and treatment plan are met. And then a doctor test chooser agent. Again, this agent was then to come up with objective classifications of what tests should or should not be ordered. And the way these agents interact with the CPC agent, which is called the gatekeeper agent, is the same way when we're seeing someone in the clinic. We get a certain bit of information, then we ask additional information, and then we learn more from the patient. So the way they structured it is that they had a gatekeeper agent that had the entire case. It would release a little bit of information, and the AI or the doctor would get that little bit of information, and then they have an opportunity to continue asking questions and get more information. So they're able to also see the chain of thought for all of these agents at the same time. So I talked a little bit about what the AI agent looked like. So there are these 5 different agents that have specific expertise. On the physician side, they had 21 physicians from the United States and the United Kingdom. These were internal medicine physicians. A majority of them were hospital-based physicians, and they were to challenge these questions and to try to address these CPC cases based on their own innate knowledge without access to up-to-date or textbooks or internet searches, which normally we would have. summary for this study is after they have they implemented both of these scenarios, the AI agent did much better than the physicians did. So the physician's diagnostic accuracy was 20%. For the AI, it was 80%. All of this at a 20% lower cost using the AI. So very interesting study, but you can already kind of see how there are some biases. Very interested to hear what you all think about AI versus physicians when it comes to diagnostic certainty for these challenging CPC cases.
My first thought is like, wow, this is impressive. And obviously there's some diagnostic dilemmas that are just really tough. And the earlier we get things diagnosed across the board, obviously we feel that way in cancer, but really for probably any specialty, the better. So that is cool. And I think there's absolutely a role. I think I would be worried a little bit about how they're accounting for bias, right? Ultimately, all of these things are built by people. The data that's getting inputted in all these AI models, which, I mean, we talk about AI and bias all the time. And that's not to say, I will also say, that's not to say that obviously humans don't have bias. And like, there's a lot of talk, like, for example, like, what do skin conditions look on darker skin, right? Like with like melanated skin, like all those things. And I'm not saying that humans are perfect at this and AI is biased. But I think I would be worried about how those biases might get kind of reinforced and exacerbated. As we know with ChatGPT, a lot of like self-validation happens within these like models. And so I don't know enough about AI to know how they're kind of accounting for that, just because like the human body is so different, I think, than any computer system, and maybe that's just my naiveness about how complicated computer systems can be, but I think biology is kind of wild and crazy. And so I would just worry about that. And I'm curious about how in the future, when I know that this is where things are going, how they're going to really adjust for that.
Well, rise of the machines, you could trace it back to 2025, right? But I think of this natural, I think as a next step of evolution, which I think is evolving naturally beyond Dr. Google, right? So, we've all experienced where people come in and say, "Well, I searched on Google and I found this," and it's very unfiltered, raw, and also in terms of quantity, an avalanche of information. In my own experience with ChatGPT, and of course its iterations and other versions of AI that have to be verified and confirmed, and layer upon layer of learning and validation will certainly be different in the future. And maybe even this same cohort, this same group with the same data, if they report it year after year, will definitely be different than what you just saw in the preprint, Shaalan. But I feel like whether patients are approaching it, I can envision some, and there's AI when you call any company a customer service, you're interacting with a human voice sounding like AI, and they're navigating you until you actually need a human being. And so the medical equivalent of that might be people saying, my knee hurts, what are the different causes? And in medicine, we call it differential diagnosis. And so if AI can parse out and maybe even a step above Google, maybe rank that might give people an indication. Now, of course, what we're talking about here with the cases in New England Journal are much, many times, many-fold, if not exponentially higher in sophistication, as well as more difficulty in terms of diagnosis, and how we in the medical community might use it. I just think of how busy we are, right? We have to go through avalanches of information. We went from having stacks of paper that are now electronic. We can access electronically, but it's a lot of information. And sometimes what worries me is am I missing a key detail? And so, as much as we train ourselves to speed read, there's so much information and you don't want to miss something. And I think there's opportunity to use these tools to go through and say, here's a key detail that takes differential diagnosis, previously number 3, and makes it rise to the top. And I'm intrigued from our side to see how that can expand. But I'm sure there are a lot of people in society that say, see, I told you that we can use computers and they're much smarter than the doctors, but I don't know that we're put out of business just yet because they still can't prescribe chemotherapy or medications.
I would be happy on some level to be put out of business if it made things better for patients, right? Like if something like really was better, like take my job, I will figure something out. But like if it actually really helps. And so Emil, I really liked your point about how much information they can kind of summarize. And I know we talked about this last podcast about how we use AI as doctors in our own lives. And I was going to say that I use the feature on ChatGPT where you can, like, instead of typing in my questions, I use the like microphone part, you know, where you like click it. Because I talk a lot, as everybody here knows. And typing just doesn't feel like enough. There's always this like one thing that I want to say, like, oh, and then I want it like this, and then I want it like this, and that takes way too much time to type. And so I'm thinking about like how even myself, like ChatGPT, when I use it, I use the microphone feature because there's so much information that I want to give to ChatGPT. And I think about like our patients, right? We, when they come talk to us, if they're talking to us for like a certain period of time, yeah, I'm taking notes, but I'm taking notes about their nausea and their vomiting and maybe how many times and maybe the like quality. But there's all these like little nuances, like, and then this happened this one time, or like this happened this like half time, or you know, and can't pick that up in the same way that I understand like you know these AI models can. And so to your point, that those key pieces might be helpful to like move on. And so I'm kind of curious about how these models perform also with people, right? Right now it's like with these New England Journal like static data that's like filled in, but it'd be and I'm sure that that's what they're doing in the future.
Yeah, I learned a lot about the agent model. So, how they designed the AI physician to have specific tasks, and they divided it into 5 different agents that all together, the sum of which will be the recommendation. So, you have checks and balances within these 5 agents that prevent anything from going rogue. I really enjoyed reading that. I'm a little disappointed that the cases that they chose were not your typical bread and butter cases that people are going to see 99% plus percent of the time in the clinic. So these were already by definition fringe cases to begin with. And then they pitted against physicians who are general physicians, not experts in the field of the cases that are there. And how often do you really have a general physician exposed to a fringe case without access to online resources, textbooks, or their colleagues as consultants in trying to solve the problem? So it really wasn't stacked in favor of the physicians. So having said that, like you said, Shruti, this is a first study that we are seeing. There are going to be many more that are coming into space, but I don't think that I'm worried about my job just yet.
Me neither. Me neither. I think there's a time. I think there's a place, but I'm happy if we can make our jobs better or easier. Better for patients, easier for us. Can't complain. Anyways, thanks for bringing that study. That's so interesting. Obviously, this is the future, and so we got to keep a pulse on what's going on. All right, Emil, you're up. What do you want to talk to us about?
Yeah, something that's been on my mind has come up in my clinical practice a lot in recent weeks and months. And it's not a new topic, but just kind of revisiting challenges associated with drug metabolism. So you know, we're practicing gastrointestinal oncologists, and colorectal cancer, esophageal cancer, and all of these type of cancers have in common drugs like 5-FU, fluorouracil. I always tell patients it's been around for more than a half a century. And there is a percentage of the population, numbering about 3 to 5% or so, that have variations of an enzyme called dihydropyrimidine dehydrogenase, which is a extreme mouthful. We shorten it DPYD. That there's less of the active amount of the enzyme, and in less than 1%, some people may have a rare variant that's either absent or near absent. And the implication for that is they have more trouble metabolizing that enzyme, and they get more of the side effects. So more what we see in mouth sores, diarrhea, suppression of the bone marrow that leads to anemia, and our white blood cells that help fight infection, and platelets that help clot blood, a lot more trouble recovering from that. And so there has been a lot of debate in the oncology community about testing, and you know, it's still at this point, why it's come up is that it's not so easy to test for it. And there's ongoing debate about, is it worth testing, is it not? In cases that are most severe, then we know it's really difficult to see patients go through things, and we have to manage that. Sometimes they have to be hospitalized or modify the dose of 5-FU for sure, if they're doing well enough that they can continue on the drug. But even getting testing in advance in any given center, whether it's large academic centers, more rural or private practices, you know, alike, just getting the testing in time for the first cycle of treating a patient like this. And so I think, you know, the ongoing debate is there are many variants of it, and not all the tests that are commercially available or even in academic labs will pick up all the variants. So it's by far not a perfect test, but sometimes a struggle of just a more common, relatively bread and butter thing. We're giving the drug pretty commonly. How do we get this test done and in a rapid amount of time? Still remains part of the, I would say, the logistical challenge of oncology care. And so, as we've been diving into the literature, it's an active area of debate, but I'd be interested in hearing your perspectives and experiences with it, because it's important for our patients to try and minimize the side effects before they even go down the road of having several months of chemo and not having their quality of life alter that much.
So I have a question for both of you. Would either of you allow a loved one to get 5FU without knowing DPD status?
Oh, wow.
Absolutely, yes, I would. But I got to tell
You
you,
would, you would allow them to get it.
I would allow them to get it depending on the clinical context. So this is the part of the DPYD testing that I think is extremely important, right? If it is if there's an urgency to get started, you have curative situation, you have things took a while, the symptoms started a while ago, and things didn't get like. I would say absolutely yes. I would say dose reduce, maybe, maybe not. Um, I think I think it's a kind of a not cost, pro con, risk benefit. That's the word I was looking for. Wow, took me a second. Like risk benefit situation where um, I actually practice at two locations. One of the locations, every patient gets the testing, every single patient. We don't start them on any treatment unless we have that testing back. And if we start them on testing, which honestly has only started happening since I got here, it's dose reduced. It's like dose reduced just to make sure that they could tolerate it for the first cycle until that testing comes back. Now, I trained at the place where it's not routinely ordered, depending on the practitioner. Some people order it, some people don't, kind of thing. And I think at a lot of academic centers, it's not routinely ordered, even though it is part of guidelines and all of that. And I guess I kind of think that I understand why it's ordered, and I absolutely think that if you can get it ordered and there isn't an urgency, if you can wait 2 to 3 weeks, I absolutely think you should. But if you can't, I think the part that I get really frustrated with at times is, gosh, if we really need to start this patient on, and it's a less than 5%, and I believe in the actual homozygous variant is even lower than that. Am I sacrificing the potential of some sort of symptom relief or management just to wait for a test? And that's like something that I think always comes up with us, whenever we're talking about anything, when we're talking about risks and benefits. And so I feel really strongly that I'm really happy we have a workflow that kind of does test for it. But sometimes there are moments where, depending on the clinical context, I do push back. And I'm like, I really think we need to get started yesterday, actually. And so, and that's, it's not a like black and white situation. And so I think if there's rules that, you cannot give 5FU if it's like mandated without getting this test back, that would actually harm patients also. But on average, do I think practitioners should feel like we should try to get it before we, if we have the ability and the time? Yes, I think it's an important test to get in the same way we get our CBCs and our Oncopay. Like it's an important test to get. I just would worry if there's like kind of mandates on it, what patients are kind of falling. I don't want to say falling through the cracks, but aren't getting kind of the care that they need in the timeliness that they need them.
And I think why, Emil, you bringing this up is so important is, I mean, the side effects are so devastating when you see someone who's truly deficient get exposure to medications. The course, the clinical course is just tremendously burdensome for that patient, fairly high mortality rate, if I may say so as well. But then at the same time, like Shruti is saying, the incidence or the prevalence of homozygous is so low. It varies across different ethnicities, and you have the urgency of starting the treatment and the cost and whether it'll be approved by insurance as factors to think about. That sort of makes us makes the implementation of this knowledge that we've had for decades very difficult to implement. And there are parallels to this to even biomarker testing for frontline biomarker testing for colon and lung cancer, for example, as well in this space.
Yeah, so I agree with so much of what Shruti said, and I think of, from the information I found, the homozygous variant as low as 0.3%, and so I generously said less than 1%, but I guess my point in the overall risk-benefit ratio, when we see about half of patients, I'm just using colorectal cancer as an example, metastatic stage 4, and of course they have definitely an emotional urgency to get started with chemotherapy, and of course there is a medical urgency as well, relatively, right? I always give the analogy, it's not Burkitt's lymphoma or leukemia, you got to get started that night, but within a reasonable amount of time, you have to get things going. One of the rate-limiting steps also is port placement and other things. So there are other factors, but if a test like DPYD might take a few weeks, I do liken it, like you said, Shaalan, to genomic profiling. If you want to know for metastatic colorectal cancer, actionable targets such as BRAF or things that make a difference in decision making like KRAS, that sometimes you just have to move forward, weigh that risk benefit, move forward to the first cycle, maybe it's just FOLFOX, and then for the genomic profiling, maybe you add the targeted agents with cycle 2 in order not to forestall and prevent someone from getting started until a month after you've met with them. And likewise with a DPYD, in my experience has been such a broad variety for the people who do have side effects that even if we never end up getting correctly the an accurate test, whether it's because the test doesn't have enough sensitivity to pick up potential variants, and so we have to speculate, as physicians in our clinical decision making, or if it ends up coming back later after they've gotten 5FU, in general, then you just naturally, what is the decision you do anyway? With or without the test, you scale back, you dose reduce, and it is trial and error. So I'm certainly grateful that it's not 50% prevalence or 25%, but I definitely don't want to diminish for the 3 to 5% that it could happen where it's less. And I've seen some patients get hospitalized, but also a lot of them will just have severe side effects after that first cycle. That just makes me think, I definitely want to confirm, I could use a test to confirm that there's a problem with the DPYD level of efficiency of that enzyme, but otherwise it's not going to change what I'm going to do, which is dose reduce. So it's hard because there's always some form of urgency in many ways to go forward with therapy. I wouldn't want to stop. So ultimately, to answer your question, I think if we play that odds, for a loved one, I think I would go forward. But of course, I would want the oncology team to be carefully monitoring as we all try to do for our own patients in real time, rather than wait 2 weeks to see what happened. I would want to hear about it as soon as possible and then make that adjustment.
Yeah,
What?
and I didn't answer the question, so I'll give mine. But
Yeah.
barring a clinical urgency, I would not let my family member get 5FU treatment without having DPYD testing, only because of how devastating the side effect. Unlike some of the other genomic tests like UGT1 testing and stuff, the side effects are not as grave as it is for DPYD. That's all.
Right. Just clarify for the audience, the UGT1A1 for the companion drug, irinotecan, that's also commonly used in colorectal cancer and other cancers as well. These are tools that we have, and so kind of optimizing workflow is a practical logistical part of cancer care that varies center to center. But it's important. I'll just add on, literally, just part of the reason why I wanted to bring it to the forefront is a commercial company that also does genomic profiling actually came out and announced that their blood-based test that's often used, that they're now using for MRD, minimal residual disease, and genomic profiling, they're now offering as part of the test DPYD assessment. And so to be able to have that as part of the comprehensive report that you would get back in ideally 10 to 14 days, really, to me, provides enhanced value. And so if that overcomes logistical concerns about turnaround time, then it's, I think, great, for companies thinking that way. I think it's very forward, and it's adding to our ability to make decisions in real time.
Yeah. All right. Okay. So last thing that I just wanted to talk about, briefly was, as we all know, there have been recent cuts to federal investment in mRNA vaccine projects. And of course, a lot of those are focused towards infectious disease. But as we all know, when basic science advances in one disease group, there's always a possibility that we can use it in other disease groups like cancer. And so I wanted to bring up a paper that was published in July of this year. And it's a basic science paper seen in mice. It was in Nature Biomedical Engineering. It's called Sensitization of Tumors to Immunotherapy by Boosting Early Type 1 Interferon Responses Enables Epitope Spreading. Fancy, lot of words, a lot of words, a lot of fancy words. Essentially, the idea behind it was that, as we know, many tumors, they don't respond well to immunotherapy. We call them kind of cold tumors. Our own immune system doesn't see the tumor as foreign and kind of attacks it. And so what their approach is they used these lipid nanoparticles, the same kind that's used in COVID mRNA vaccines, to deliver RNA to the body. And the RNA actually wasn't tumor specific. It was like a generic danger signal that revved up the immune system, again in mice. But the results in mice were that tumors that were previously resistant to immunotherapy started responding. And the immune system learned to target multiple tumor antigens and not just the original ones. And so I kind of, and I'm sure you both know about kind of how we have a little bit of excitement on the forefront for how we can use vaccines in cancer. And so just wanted to bring it up and see what your guys' thoughts were on that. I think, funding basic science that doesn't seem relevant to cancer care specifically, but might be able to be used in the future in cancer care is important and relevant to us as oncologists.
I think vaccines are an extremely exciting area for cancer treatments, whether that's the mRNA vaccines or neoantigen vaccines or their peptide-based vaccines. The way we have seen other forms of immune therapy develop in the cancer space, especially in those who have low volume disease or in the adjuvant space, there's a lot of room for us to exploit the immune system in ways to help improve cancer control. Having said that, to the point I think that you made, Shruti, in terms of the deprioritization of a specific type of investigation is going to slow down innovation in that area. But we've seen this before, and I'm not saying whether it's right or wrong. We saw this with stem cell research in the early 2000s when, because of political pressure, an entire area of investigation was deprioritized and defunded, and that pretty much ended a lot of innovation in that space without really judging whether we're better off or not because of it. I do think continuing to learn from the basic science discoveries that we have, it may give us very interesting mechanism-based insights that doesn't directly relate to the use of mRNA specifically in humans. It could give us specific pathway-based, mechanism-based information, which we can use for our other treatments or can inform patient selection or treatment selection downstream. And that's what makes the basic science work so important, is when you're starting off in space, in that area, in that specific area, you don't always know where it's going to go and what the subsequent consequences are going to be. We are all translational/clinical investigators. We're on that end of the spectrum, right? And sometimes I think we lose perspective of the importance of just hardcore discovery in the basic science space, and to see any area be smothered for whatever reason, can have downstream consequences.
That's a beautiful example of cross-pollination, bench to bedside, is an often used cliche, but cross-pollination between disciplines, right? So here's an example of cancer research and translational application in the immunotherapy sphere, attacking a very relevant problem. So, as you mentioned, Shruti, the cold and so-called hot tumors that immunologically and immunotherapy responsive versus not sensitive to yet. There's so much of active research trying to convert unresponsive to responsiveness to immunotherapy. But the next frontier is figuring out how tumors become refractory, right, and then no longer respond to immunotherapy. And this mRNA, research really, rose from the ground up, and obviously during the COVID era, the vaccinations that led the way, really based on mRNA-based discovery. I think we should take the opportunity here to educate the public what mRNA is, because I think it's just unfortunately wrapped up in so many other things outside the scientific and medical sphere and misunderstood and unfortunately been complicated. And mRNA is really just genetic material derived from DNA, and it's kind of the intermediary path to proteins. I find it fascinating just from a vaccine standpoint, whether it's the cancer vaccination approach or vaccination for infectious disease, that the old paradigm would be you used to have something on the cell surface of a virus particle, or in cancer, a cancer cell surface protein type that had to be the target to create vaccines. But to have mRNA be something immunogenic to create vaccines of any kind is just fascinating. And to have it as a delivery vehicle for something like this to help it is as you said, of course, it was in mice, so not yet ready for prime time, but this is the way station to get to that, and that's really important work. So mRNA, I think it's my great hope for 2025 is that this doesn't get caught up in the political zeitgeist and the funding that should go into this, and if it does not in 2025, we'll have repercussions not just for infectious disease, but for cancer care. Direct clinical trials that could lead to standards of care in the years and decades to come would be forestalled and delayed significantly if any of that is interrupted in 2025. And you could say that about so many things about basic science research, but the immunotherapy of today that people are clamoring for, right, whether or not their cancer type, is eligible for it, the seeds were sown in the 80s, 90s, 2000s, and then Dr. Allison, who won the Nobel Prize for discovery of PD-L1, he said the story many times. He wasn't intending to come out with the next great cancer treatment. He was doing pure basic science discovery, but it took years for that to blossom into the great advances of immunotherapy that we know today. And now part of the next frontier is figuring out how do tumors evolve against immunotherapy and how to overcome that. And studies like this are really important.
Absolutely. I think another really good example of this, unrelated to cancer care, that I think people talk about a lot is the Ozempic and all of the diabetes medications, but now for weight loss, came from discoveries from the Glia monster venom, which is like not Glia monster. Yes,
Right.
Glia monster. I almost was like, did I say that right? Because it's what a wild name, the Glia monster, but it's like a venomous lizard. And they studied one of the hormones in the Glia monster, and now we have not just a diabetes medication, but a weight loss medication that's showed cardiovascular benefits and I think benefits in sleep apnea. And so for something that probably, I don't know that they knew that this is it's going to revolutionize care for quite a few people. All right, guys. So that's all the time we have today. Thanks so much for listening to us. We love talking about everything science related and how it's related to all of our daily lives. So catch you guys next week.
Bye.